Tidal saline wetlands are highly productive ecosystems that store a significant amount of carbon, representing a critical component of the global carbon cycle. Salt marshes are one type of tidal saline wetland. With sea levels projected to rise, tidal saline wetlands have the potential to provide a negative feedback in the climate system by storing additional carbon. However, the expansion of these salt marshes may not be able to keep up with the rapid pace of sea level change. Dr. Tanner's marsh coring research is part of a pilot study to analyze the effects of changes in the extent of different marsh types on the rate of carbon sequestration. Dr. Tanner plans to radiocarbon date the sediment at the base of 9 cores taken from the marshes northeast of Pond Island in the Schoodic section of Acadia National Park.
What we're interested in doing is figuring out the rates of carbon storage in these different types of wetlands that we all have right here to see which ones pull in the most CO2.
Last week, Sarah and I went out in the field with Dr. Tanner and his research assistant Lee Sorrell to see what the coring process looks like. Because we needed samples of freshwater, low and high marsh sediment, it was important to learn how to distinguish the different types. Marsh type is determined by the dominant plant species. The different shades of green and the varying heights of the plants across this wetland are indicative of different levels: freshwater, low, and high marsh.
Then further back is what we call higher high marsh. You see the vegetation change back there to the Juncus Girardi. And we also have a little bit of low marsh on the other side of the road here. And way back there, kind of past the little pond you see there, is a freshwater wetland.
The extent of each type of marsh was mapped using a GPS unit. We selected 2 points within each marsh zone for a total of 9 points and recorded the exact location of each with the GPS unit. A water quality probe was used to measure the pH, oxidation reduction potential, or EH, and water temperature at the different marsh sites. At each point, a series of 0.5-meter long cores were taken with a Dutch auger, a manual hand coring device. To extract a core, the auger is shoved into the soft marsh ground as far as it can go. Gripping the handles, the auger is twisted and pulled out of the ground. Encased in the auger is a cylinder of marsh sediment. The cores are described by color and composition. Colors are named according to a Munsell soil color booklet that contains swatches of possible soil colors. This naming convention makes certain that researchers in other countries can look up the color name listed in Dr. Tanner's study and the Munsell booklet and find the exact match. Composition refers to the presence of organic matter and any irregularities. This core is described as having roots throughout. Cores are then sliced for subsampling at 5 centimeter intervals. In the lab, these samples will be tested for percent carbon composition. Sediment samples from the base of the cores will be sent to a separate laboratory for carbon-14 dating. Once determined, soil carbon accumulation rates will be compared for the different marsh zones. Knowing how fast soil carbon accumulates in each type of marsh informs researchers of the effects of marsh transgression on the carbon storage potential in these wetlands.